morning, everyone. Good morning, everyone here this morning. Hudson River Foundation, and welcome all of you that are on the webinar. I think we have about 15 people here in our office, and I think there are about 40 or so of you on the webinar. Just as a little bit of background for those of you that are viewing this on the webinar, hopefully you can see us and you can hear us. We can't hear you, although we'd like to, but at the end of the seminar, you'll have an opportunity to ask questions of our speaker. I think you've got some directions on that. You'll have to write them out, but we'll make sure that we get to them once the talk is finished. This is our first Edward Ames seminar of the winter season, and we're pleased to have Edward Ames here in person. Thank you, Ned, for being here.
Thank you, Dennis.
And sometimes over the years at the foundation, a lot of the work that the research that we've sponsored, we like to sort of break them up into different components, looking at dynamics of food webs, looking at hydrology and biology of species. And we funded quite a bit of work on contaminants over the years. And in general, we've looked at the fate of contaminants that enter the Hudson and the harbor, and look at what happens to those contaminants, how they may change, where they ultimately go. And also we've funded work on the effects of particular contaminants. And it's somewhat unusual or rare that we have projects that look both at the fate of contaminants and also the effects of contaminants. And over the last couple of grant cycles, we've been particularly interested in learning more about some of the contaminants of emerging interest. And I think that probably those sorts of terms, we've been looking at contaminants of emerging interest for so long, they're not emerging anymore. They're here, they're with us. But there are certainly compounds like personal care products and pharmaceuticals that we haven't really looked at the fate and effects of those to any great degree in the Hudson and the harbor. And we were pleased to get a proposal from Max Häggblom a couple of years ago that we were delighted to fund, and he's going to talk to us about some of the results that are emerging out of that particular work. Max is, those of you that may not know Max, he's a professor and chair of the Department of Biochemistry and Microbiology at Rutgers. I guess he's been at Rutgers for about 20 years, doing all sorts of wonderful work on microbial ecology. And as the talk talks about bugs on drugs, he's going to tell us about how bugs may be altering these particular pharmaceuticals and personal care products, and give us some insights, preliminarily at least, as to whether some of the changes that go on caused by these bugs may be good for the environment or maybe bad for the environment. We shall see. So, Max, thank you.
Well, thank you very much for the invitation to speak here today, and I also want to thank the Hudson River Foundation for many, many years of funding on different projects. I think over the last 30 years now. So when I first came to New York University as a postdoc was really young. One of our first projects was actually on Hudson River Foundation funded, and then on projects further. So yes, this is really going to be talking about emerging data on emerging contaminants. And as an environmental microbiologist, I'm interested in what I call unusual appetites. So how microbes have evolved, have adapted to degrade different toxic chemicals in the environment, and especially so-called anthropogenic pollutants. Again, we worked on Q-oxygenase, brominated flake retardants, dioxins, PCBs, also interesting metalloids, and so on. And one aspect of our work is always, once we figured out how one contaminant might be degraded, is of course then to start examining the next one. So what is then the next emerging pollutants? And of course, over the last 10 years or so already, there's been more and more interest and concern about this range of chemicals called pharmaceuticals and personal care products. And so these are really a diverse group of really hundreds, thousands of different chemicals, different structures, and they're used, again, as prescription drugs, either for over-the-counter drugs, we have veterinary medicines, and then also fragrances, cosmetics, and so on. And so it's a really bewildering array of chemicals, or wonderful array of chemicals if you're an organic chemist. And they've now very clearly been established as contaminants of concerns in watersheds around the world. They are released into the environment either directly or indirectly, that their long-term fate is poorly understood. Many are biologically active, and of course, especially the drugs, so they have some biological activity, and again, depending on the chemical, can have adverse effects on aquatic biota. And so therefore, we've made the argument that it's really important for us to understand the fate of these chemicals, how they could be degraded, how they could be transformed, and again, that's been the goal of our work. And just a slide where I know you will not see any detail whatsoever, that's actually the purpose. This is data from studies by the U.S. Geological Survey, published in Environmental Science and Technology a number of years ago, examining a range of different watersheds in the U.S. and just listing these various pharmaceuticals, personal care products that were detected. These include veterinary and human antibiotics, prescription drugs, non-prescription drugs, other wastewater-related compounds. I'll talk a little bit about bisphenol A, which sometimes gets lumped into the same group because it behaves similarly. And of course, then a whole bunch of fragrances, shampoo components, and so on and so on. So these are detected really just about in any water sample you take. There's a lot of data now coming up in terms of concentration ranges, detection points, and so on. This is just some of the data from that same study, examining steroids, other drugs, disinfectants, plasticizers, and so on, at what concentrations they were found, how frequently, and then also the number of different compounds that were found within this class. So again, we have really an amazing array of compounds, and of course the concern is then not what's happening to these compounds and what are the effects against fish, other biota, and of course also eventual exposure to humans through the reuse of the water. So we need to understand partly what's going on with these PPT compounds coming from domestic use, from industrial production, from hospitals where they may be in much higher use, also in animal farming, agriculture, so aquaculture as well, where in all of these cases, many, many different pharmaceuticals are used. One of the biggest concerns is, of course, antibiotic residues and the increase in antibiotic resistance genes. That's a separate question that I won't go into today. But then, what are the different routes of contamination into rivers, into groundwater, and so on? So understanding, again, how the different sources, what are the different sinks, what is then happening to these chemicals once they're into the environment. So we really have also a question of what we can now call an urban water cycle. Thinking of first, what are the different freshwater sources for the community water withdrawal, what might be input into this initial source, how is it purified, how is it used, what is then the treatment of wastewater, and where is that being discharged, and then how is that river water, for example, reused again. And this gets into really what is this broader question into microbiology and that of xenobiotic compounds. So chemicals introduced really in what we really can call the Anthropocene, so the last 100 years or so, these chemicals have never existed on Earth before we started producing them in industry. So completely new chemicals. The question then is, how might microbes be able to handle them? Can they degrade them? Or do they convert them to other compounds? And so what we call about the transformation, so also various metabolites. And so again, that's the driving factor in our work, is can we understand what microbes how they might use these chemicals, what might be the end products, and what are the effects of those products again. So again, understanding and linking them with human medical use, veterinary medicine, what are the different disposal routes, excretion, manure use, or sewage, what goes into landfills, how do they leach out of those, again, what is going through into our drinking water, into groundwater sources, into the food chain as well as these chemicals. So these are the broader questions. And then much of this also has to do with thinking of our wastewater treatment. So most of the pharmaceuticals that we take are fairly water soluble. They are about 90 to 99% excreted in urine. So whether we would pour them straight down the drain or use them first as prescribed by our physician, we're peeing out most of that chemical. Ends up in our sewage, and then the question is it degraded in our typical sewage treatment process? And for the most part, that answer is no. To some extent, but there are very much data now showing quite a lot of the residues coming through. And part of this, of course, has to do how a typical sewage treatment plant has been engineered and really emerged to degrade readily degradable organic materials. So we have our secondary biological treatment, or typically it's an activated sludge treatment plant, a hydraulic retention time of 6 to 8 hours or so. Excellent for degrading all of the fats, sugars, anything else that are essentially readily degradable organic molecules. Anything that is more recalcitrant is not going to be degraded through this high throughput system, and depending then on its water solubility, either going into the discharge or in some cases sorbing onto the sewage sludge and then being removed with the sludge, and then it's the question of how that sludge is treated later, whether that chemical gets back into the environment. And this is really a question of what's called microbial infallibility. This is a concept really developed further by Martin Alexander, one of the pioneers in biodegradation, with this thinking of what's going on with the global biogeochemical cycle. So any organic molecule that's naturally produced, somewhere on Earth, under some conditions, there's a microbe that can use that chemical as a food source. And why do we sort of know this? Well, if that would be the case, we would be up in our eyeballs with that chemical by now. And so, again, most organic molecules then fall into our understanding of the carbon cycle. But what happens now when we have new, novel xenobiotic compounds? So chemicals that are completely foreign to the biosphere, microbes have only seen this for the last 100 to 200 years. Has there been enough time for evolution of new degradation pathways, evolving of new microbes that are now seeing this new organic molecule as the food source? That's really, as a microbiologist, my main question. And that, of course, comes into this question of the eventual environmental fate of the chemical. So depending on then its degree of degradability, we can classify them as readily biodegradable, they could be persistent, or then highly recalcitrant. Now that is very much a relative term. Depends on, so this persistent depends on the chemical, its structure, its chemical properties, the particular environment. Are we looking at a aquatic system, a sediment, a soil? And of course then the time. Are we measuring persistence in a few hours, such as a wastewater treatment plant, a year, a decade, a century? So again, it's very much a relative aspect of what we consider a persistent chemical and what is not. But again, if it's recalcitrant and if it's introduced into environment at a rate higher than which is degraded, of course, it's going to start to accumulate. And we have a whole bunch of legacy contaminants, our so-called POPs, persistent organic pollutants from PCBs on, that it's well understood that yes, we have problems. The question now, what about some of these different PPCPs? Again, some are clearly shown to be degradable, others we already have data that they're recalcitrant. So this, again, we need to realize that when we're talking about pharmaceutical personal care products, not one chemical group, we are really hundreds of different ones. And then from our microbiological questions, so thinking of the different sources, sewage discharges, stormwater runoff, and so on, what's happening that's partly just chemical based, how is it moving in the watershed and so on. But then, of course, what's happening in sediment, the biologically active zones, so the aerobic areas, but also in anoxic systems of the sediment, depending on then where that chemical starts to accumulate. So where we've started our work is partly from data that's already established that a lot of the PPCP compounds are released from wastewater treatment plants essentially untreated. To some extent they may be degraded, but we're seeing in effluent increasing concentration. Once they're discharged, we actually know very little about how they are degraded, where they are degraded, and what are the rates. Our overall hypotheses are really one that microbial transformation of these compounds will generate downstream metabolites that can then be degraded further by other microbial community members, but also have various effects on the biota. So the question of toxicology comes into not just the parent compound, but also the metabolites. What we also think is happening, and there's of course evidence from other xenobiotic compounds, that once you start to introduce this new chemical, there is a bug somewhere that realizes, hey, I can eat this. And if I can eat this and my partners can't, well, I get the competitive advantage. This is food only for me. So there is immediately a selection pressure for evolution of new biodegradative pathways to start degrading whatever new organic molecule is released. Again, if you can oxidize that completely to carbon dioxide and get energy out of that process, this is now a new food source. And again, one of our questions then is how can we start to monitor that process? Now, I'm particularly interested in sediments, in anoxic systems. And again, that has to do with my previous interest in a lot of hydrophobic contaminants. And again, some of the PPCPs also fall into that class. And so once they are entering an aquatic environment, if they're sorbing onto particulate material, that's going to sediment out of solution and very soon get covered up and end up in an oxygen depleted zone. So one of our broader questions is understanding how different electron acceptors can be used by microbial communities for degradation. And that's, again, we're using oxygen to respire. Microbes can basically respire virtually any other molecule that has different redox states. Some of the very common ones, of course, are nitrate and sulfate. Sulfate is, of course, the key electron acceptor in marine systems or estuarine environments where, again, there's 30 millimolar sulfate in seawater. So this is a very abundant potential electron acceptor for microbes to use. And so our question here is, if we're looking at the oxidation of organic molecule, here is just shown acetate as a simple example. It's being oxidized to carbon dioxide. Well, that oxidation has to be coupled with the corresponding reduction. So can that oxidation be coupled to reduction of oxygen to water, aerobic respiration, nitrates to nitrites or ammonia in the process of denitrification, manganese and iron oxide reduction, sulfate reduction to sulfide, or in also the process of methanogenesis. And so again, these different electron acceptors, we also need to note, are not created equal. There's a difference in their oxidation reduction potential, so microbes get different amounts of energy if they are respiring oxygen versus sulfate. And that also affects the rates of degradation and rates of growth of the microorganisms. The other aspect that is interesting is that there's a very distinct zonation of these redox habitats, especially in sedimentary systems. This is actually one of our early sediment cores from Vestry Street on one of our early projects on PCBs with Richard Bach, who helped us collect that. And it's a wonderful picture that I keep reusing because it very nicely shows this zonation that we see in an estuarine environment. The top sediment is light brown, again, air oxidized, but as you go down a few centimeters, turns black, mainly because of the production of sulfide that precipitates with iron into this black precipitate. So, and you also find the smell of hydrogen sulfide on there. And depending on then the sequential use of these different electron acceptors, we have different zones of processes. On the top, there's aerobic respiration, then we will have a zone of denitrification, then manganese and iron reduction, and eventually down at the bottom, a strong zone of sulfate reduction. And when all of these electron acceptors are utilized, we have a zone of methane production, methanogenesis. So a chemical, whether it's a PCB or now a personal care product that sorbs onto our particulate compound sediments here, collects over time as more and more sediment accumulates, it's going to travel through this redox zone. If it's not degraded in the first couple of months, it's then going to be in a denitrifying area. After a few years, it will be in a zone where iron reduction is important, and eventually where also sulfate reduction predominates. And so this is why we're interested in understanding how different electron acceptors can be used for the degradation of these sets of chemicals. So one of our key questions really is then to understand how the redox environment impacts biodegradability, and especially in an estuarine system where you have a strong input of sulfate through for the tidal influence. And again, can we identify the microorganisms that have this capability for degradation? Can we identify the functional genes, understand the pathways, and of course then also develop the different monitoring tools for degradation and fate of PCCPs. So our specific task is again examining how the redox environment impacts biodegradability. Can we combine a technique called enrichment culture with then different molecular tools to identify microbial community members to understand who is active, who is growing on their And later on, with some collaborators, I'm not going to go into this in detail, can we also develop other analytical tools such as stable or compound-specific isotope analysis to understand the fate of select PPCPs. Now, the concept of an enrichment culture dates back really to the early days of microbiology. Two of the pioneers in microbial ecology, Sergei Vinogradsky and Martinus Weyrink, developed what is called as the enrichment culture technique. And basically, it's based on the concept that the microorganism that grows best under a specific set of conditions is going to outcompete others and become predominant. So, Bob Becking, one of Weyrink's students, said everything is everywhere the environment selects. So if you have the right environment, the organisms, bacteria can travel all around the world over thousands or millions of years. They're eventually going to be there. If the environment is right, they're going to grow. And so we are specifically then focusing this on, can we find organisms that can degrade this select group of PPCPs. We're focused on the painkillers, aspirin, acetaminophen, ibuprofen, diclofenac, partly because of our interest in aromatic degradation, but also these are among the most prevalent PPCPs found in sewage effluent, as well as both now the Hudson River, the Raritan River, where we've done analysis. So these are common chemicals. We're using them, and so our basic mode of action is we will go to a site of interest. This is from New Brunswick, the Raritan River. One of our favorite sites is actually the Arthur Kill, because it's been exposed to every possible chemical on Earth. And this is already with Lily Young, who I've worked with. Our basic hypothesis that if it's not degradable into Arthur Kill, it's not biodegradable, period. And so far, that's helpful. We can then take some sediments back, set up microcosms under controlled conditions in the laboratory, add different electron acceptors, spike it with a chemical of interest, and now start to monitor how that chemical is broken down. One of our other sites that we're working on, I'll show some data from the 79th Street Boat Basin in New York. Again, what I'm looking for is typically a simple place where I can get a jar full of mud to bring back into the laboratory and then start studying this microbiology. Here, we are working and focusing specifically on strict anaerobic microorganisms. There's been quite a lot of work on degradation of these compounds by aerobes, so organisms that use oxygen for respiration. We focused our work specifically at what's happening in the anoxic sediment column. So one of the issues that we have to do is, of course, keep oxygen out in all our manipulations. And there are different ways of how we can generate microcosm under a headspace of nitrogen, seal that, and then spike it with our chemical and monitor. And we can then sample on the bench using syringes that we can go back into our microcosms and keep them active and study them for many years if needed. So here's our initial data where we started using Arthur Kill sediment microcosms as our start. And so in this case, we're spiking individual sets of microcosms with either aspirin, acetaminophen, diclofenac, or ibuprofen. We monitor these under 3 different redox environments. So we're adding sulfate to about 20 millimolar to drive the microbial community towards respiration with sulfate as the electron acceptor. In another step, we're adding nitrate, again, roughly the same amount, 20 millimolar, where if there are denitrifying microorganisms present in this sediment sample, they should now be able to use this organic molecule. oxidizes to CO2 coupled to nitrate reduction. Or if we leave all other electron acceptors out, these will eventually or can be degraded coupled to methanogenesis. That is basically the lowest redox point where then methane is the end product of organic compound degradation. And then we're monitoring over time, taking samples. In this case, we're spiking a relatively high concentration, this is an order or 2 magnitude higher than what we would find in the river water or the sediment. But again, this is for us to tease out a potential microorganisms with this metabolic capability. And as you can see, aspirin, acetaminophen is under slightly different rate. This is over about 2 to 3 month period. We're completely depleted out of these microcosms. I'm not showing the sterile controls where we know it wasn't just disappearance. If microbes were not alive, there was no loss of any of these compounds over essentially a 1 year period and so on. And there's some differences between the redox environments. Some go faster under methanogenic, others under denitrifying conditions. Sulfidogenic conditions appear to be so. But again, diclofenac is my favorite painkiller, ibuprofen, not degradable. And so I'm starting to think of, well, actually, should I really be switching my use on there? Because that just shows that once ibuprofen ends up in sediment, it doesn't seem to be going away. And we monitored these now for a year, and we're still seeing no loss. Why? There's not that many other side chains or so on. So that's the interesting question for us, where we can start to be doing also structure-activity relationships. Why is one aromatic acid degraded and the other with a few other side chains not biodegradable? That very same overall picture also comes across from a little bit upstream, the 79th Street, so both basins. So this is our Hudson River microcosm. Again, aspirin very quickly degraded, the same with acetaminophen. No loss of either diclofenac or ibuprofen under these conditions. What we are now doing is, since we see loss, the next step for a microbiologist is, well, can this degradation activity be sustained? Are they truly using aspirin, acetaminophen as their carbon and energy source? If that's the case, we should be able to respike the molecule and it should be degraded even faster, essentially showing that there's growth of the population. And we should be able to transfer the culture into fresh medium, respike the compound, and again see the community kick in and start to do it. And we've been able to do that. We've essentially now enriched what multiple fold the microbial community. So we have stable enrichment cultures that are using either aspirin or acetaminophen as their only carbon source, coupled to either nitrate reduction or sulfate reduction. What we're now working on, and I don't have the data yet to share with you, is who's doing it. That's the next step for the microbiologist, is figuring out what different species are active, and, for example, comparing the Hudson River with the Arctic Hill, with sites in the Raritan that we're working on, and also a site in China, the Zhuang River near Xiamen. Is it the same organism that's doing it everywhere? So is it, or do we have variation in the microbial community? That's a question then when we're trying to figure out and develop monitors for biodegradation as well. So we're also expanding now our work to use the Raritan River watershed as a model, and there's 2 reasons for this. So we've collected sediment samples, water samples from the bay upstream. New Brunswick is right here, and what we have here are essentially 2 different gradients. We have a redox gradient with the tidal influence and, of course, introduction of sulfate-rich seawater into the lower Raritan. And then we have a likely P2TC gradient. There are multiple wastewater treatment plants, of course. This is a highly urbanized area, but as we go upstream, we should have cleaner and cleaner water where the microbes actually have not yet been exposed to high doses of these different compounds. We'll see. We're still working on that data. I mean, I guess you're not expecting us to find any clean place anyway. Yeah. We may need to go farther up into really remote areas. But that's, again, our question is, one, how does the redox environment affect the growth, enrichment of degrader populations? But then also, is there anything that we can link to previous exposure to these compounds? This Rutgers just has a wonderful new sampling boat that's available for use, and so that gives us very easy. We can go to different locations, get a grab sample of top sediment. We've done also a lot of initial water analysis where we're doing solid phase extraction, and then identification and quantification by liquid chromatography, mass spectrometry, where we can get an idea of some of these compounds. And this is actually the data from this set of the lower Raritan, so basically from New Brunswick down to the Bay. What do we see? Every other person is drinking coffee. Caffeine, yep, great monitor of sewage effluent. But then we have aspirin, acetaminophen, still as major components in here. Also, that's a cardiovascular drug that I'm still trying to look at why that is so high versus others. But again, this is one way of yeah, everybody seems to be having aches and pains, and these are water soluble and get in. Interestingly enough, ibuprofen is not on that list. Well, actually, ibuprofen is not that water soluble. It will sorb to the sewage solid and probably is removed that way. So there's probably a 50% reduction in the sewage treatment plant that is most likely just from sorption. And it also, since these are surface water samples, well, we'll probably see ibuprofen more in the sediment layers. Now, our data is showing, well, if there's no oxygen, they're also not going to be degraded, and so that's going to be one way of how this data can then be used to start thinking about the long-term fate of these different molecules. We also set up a slightly different type of experiment, and this is one to mimic the sewage overflow. Again, with every major rainstorm, anything that goes straight through untreated is into the river. So we actually mimic that. We took our Raritan River sediment, and then we, with a sediment river slurry, added about 10% of wastewater treatment plant influent from one of the New Jersey sites, and then incubated these over time. This shows in blue our time 0, and our second time point was 4 months later, showing, and again, there's a range of the compounds that are protected. We can see some are degraded, others are not. But for us, this is a great way for us to be able to actually much better mimic what's happening, and also, instead of looking at one chemical at a time, getting actually this array of compounds that are produced. Again, I just got this data Thursday from my students, so I haven't actually been able to analyze all of the details in terms of what's degraded or not. But again, there are a few where we're seeing significant losses, but others are actually still present there even after 4 months of incubation. Now, the other question we had was about metabolites. So, just hitting back. So here, when we're studying aspirin, acetaminophen, and we're refeeding. We're not seeing anything accumulate. There's essentially destruction of the aromatic ring. We're seeing growth of the microbial community. So in this case, the likely explanation, and this is what we still need to confirm, is that this molecule is being broken down completely to carbon dioxide. It's being mineralized. But what happens when it's not? And that question we've studied earlier with bisphenol A. That's, of course, the molecule well known from all our plastics. This might already be a BPA-free bottle, I'm not sure. And work that we did in understanding how, in some cases, microbes degrade a chemical, so they basically tear apart the chemical structure. In other cases, they transform it, they metabolize it into something else. And what we observed was that a number of bacteria will slap on a methyl group to the hydroxyl. So our parent compound EPA, we could observe, was over time transformed to two metabolites that we could identify by GCMS that accumulated over time and were not degraded further. So we see the increase of either the monomethyl or the dimethyl ether derivative of bisphenol A. So what? Should we be concerned? Well, we already know that bisphenol A is an endocrine disrupting compound. It binds to the estrogen receptor, not as strongly, but again, this is one of the main concerns with BPA residues. Well, what about these new metabolites? This is where we teamed up with Keith Cooper and Lori White in my department at Rutgers, using the zebrafish as a model to study the effect. So basically, as a microbiologist, I'm looking at, okay, we have a microbe that does something to a chemical. Can we identify that new metabolite and now start testing this compound for its potential effects? It's a simple model. We can have a large populations readily in the laboratory. It's sensitive to many compounds. It's an excellent model for toxicological studies. And again, we can also extrapolate the results to human and to broader environmental health. The zebrafish genome is now known, and so one can develop a whole bunch of assays for specific enzymes involved in the life development as well to monitor activities. And one of the first things we studied is toxicity. So you can feed bisphenol A, the monomethyl ether, the dimethyl ether to zebrafish embryo, and then you monitor basically how many died. And you can get lethal doses on there. Again, both the monomethyl ether, dimethyl ether have at least threefold lower LD50s than bisphenol A. Yes.
Yeah. Is this the BPA that mimics estrogen?
Yes.
Okay.
And then the question, what about when you slap a methyl group or two methyl groups onto that hydroxyl? One of the things, addition of a methyl group makes this compound more lipophilic. So it's actually going to stick more in membranes. It's probably going to accumulate to a larger extent than BPA. And the question is, does that also impact the toxicity? And again, the other aspect that we looked at was observing different types of lesions, abnormalities in embryo development. And so this is again why the zebrafish is such a great model, because you can have multiple embryos, monitor them, register for edema, hemorrhage, and then compare the different molecules to each other. So what we saw from this work was that, okay, BPA is methylated, so the hydroxyl groups are methylated readily by a set of bacterial species that we studied, but this metabolic capability appears to be fairly widespread in different microorganisms. And what now, after we have published this work, now a number of investigators are looking for the mono and dimethyl ethers in wastewater, in receiving waters, and are finding them. So clearly it's already shown that this methylation occurs, for example, in a wastewater treatment plant. Before, I mean, if you're not looking for this new molecule, you're just searching for BPA, it may be gone, even though all of it has been converted to a derivative. Now, they are more toxic. They also have, or start to activate a different cascade of the estrogen activity, and this is one where we still don't know the details of those activities. But again, clearly, these metabolites are not innocuous, and we now need to understand their fate in the environment. And of course, with all the discussion about BPA and concern, now of course, just about any plastic bottle or container on the market is touted as BPA free. They're still, I think, mainly in the linings of tin cans. There's not a good replacement, so there is probably your biggest exposure today to bisphenol A. Of course, also from the receipts at cash registers, they use BPA in the ink. And what do you think is BPA free then? Well, they're now using either bisphenol S or bisphenol S in the plastics production. BPA, there is some positive aspects, yet these are not as strong binders to the estrogen receptors. So their ecotoxical effects are definitely lower. But our question, very similar to the hydroxyl group of BPA that got methylated, we're now examining. I don't have yet the data. My student is working on it. What about bisphenol S? What about bisphenol S? Are these also methylated? And in that case, how do these new metabolites behave? That's, of course, the key ecotoxicological question. And nobody's doing these assays yet. They're just studying the parent molecule. And that has been my argument all along in terms of understanding, yes, microbes will transform chemicals to new metabolites. We need to know that whole chain to truly determine the concern with a given chemical. Now, it becomes even more interesting in some respects. We've also worked on one of the most widely used flame retardants, tetrabromobisphenol, and you can see how this is now connected. It's the same carbon structure, and you're probably sitting on it. Tetrabromobisphenol A is used in polyurethane foams to suppress fires, and so it's in our computers, it's in the furnishings, and so on. And we've shown that this compound also is methylated to the mono and dimethyl ethers, actually even more readily than BPA. For reasons that I still don't understand, if you have halogen substitution on both sides of the free hydroxyl, it seems to be a magnet for methylation. It's like 2 orders of higher activity than the free BPA molecule. So these are very readily methylated. And again, we've shown also earlier with similar work with zebrafish that, yes, these molecules are also of concern. Now, in an anoxic environment, there's another process that my laboratory has worked on for many, many years, and that is reductive dehalogenation. This is actually another very interesting way of respiration that microbes have developed. So instead of reducing oxygen to water, these bacteria reduce a halogenated compound to a non-halogenated one, and couple that with the generation of proton-motive force in their membranes, which is what we call respiration. So this is the process of respiratory organohalide, dehalorespiration, respiratory dehalogenation. It has probably half a dozen different names on this. And we can find these organisms just about everywhere. They're in the Hudson River sediments. They're important in dechlorination of PCBs, of dioxins, and also in the reductive debromination of flame retardants such as Häggblom or bisphenol A. Now, what is the product of that debromination? Well, that's bisphenol A again. So we have bisphenol A coming from a completely different source, the use of flame retardants. And again, if we look at what's happening again at this oxygen anoxic interface, aerobic bacteria mediate this methylation. In the anaerobic zone, we see reductive dehalogenation. And so far, we have seen no evidence of anaerobic degradation of bisphenol A. Some of our cultures we monitored for 3-4 years, no loss whatsoever. So BPA, phenol can readily be degraded. This more complex molecule, we see no evidence of anaerobic degradation. So if it's in sediment, it's going to stick there. Aerobically, there are a number of bacteria that can utilize this. So it is biodegradable. But clearly, again, persistence depends on the chemical, the time, but also the environment. So presence, absence of oxygen, presence, absence of other electronic sectors becomes in the system. So what we've shown is that some pharmaceuticals and personal care products can be degraded to various degrees in aquatic sediments by native organisms. Others are not degraded. Depending on the dominant redox condition, since there's presence, absence of different electronic sectors, the biodegradation rates and extents are going to be different. We've seen actually with our small subset of compounds, very similar degradation profiles at every single site we've studied, both here on the Hudson River, the Raritan River, also the sites in China. Again, so these organisms with this capability appear to be globally distributed. Are they the same species? That we still do not know. But again, other PPCPs can be transformed to metabolites with potential adverse biotic effects, and I'm lumping bisphenol A into this group, even though it's not strictly a pharmaceutical or personal care product, but it is actually in our shampoo bottles where we have our personal care products and actually leaches out with them. So it behaves very similarly to these others. And clearly, there is much we still do not know and need to understand on this. So again, the question from a microbiologist is what's happening with biodegradation versus biotransformation? When is the molecule completely torn apart to carbon dioxide? When it is transformed to new metabolites that may then accumulate in the environment and have very different effects in the parent compounds. So to get us to this point, I've had a group of wonderful students and postdocs who worked on the project. Amani Rupakula, who's was a postdoc in my lab, Michelle Sellis, a current PhD student, and Jessica McCormick, a former PhD student who worked on the bisphenol A project. A set of really eager and excited undergrads, Alex, Albert, Sam, and Shivani, who are currently working on different aspects of the project. Collaborators at Rutgers, especially Lee Kirchhoff on our community analysis, and Lori White and Keith Cooper on the toxicology. some of our analytics, we're teaming up with STEMU at the Chinese Academy of Sciences Institute of Urban Environment in Xiamen, who's also working on PPCP compounds, and so we're teamed up on some of the analytical work and hoping to get in with Ron and I and Hudson Huntsley on the question of how we can go a step further and use carbon stable isotope compound analysis to study things. And of course, something from the Hudson River Foundation, also the Raritan River Consortium, and the Rutgers Center for Global Advancement in International Affairs. So happy to answer any questions.
So you bring up the idea that wastewater treatment plants could have a terrestrial treatment of waste by with soils, filter the wastewater treatment water after it's processed.
That's true. I mean, again, for all of these chemicals, it's going to be the initial question of water solubility, absorption onto the tube solids or not, or then very, very rapid degradation in just an 8-hour period. Clearly here we're looking at a different time scale for the anaerobic systems. So something that's readily degraded by aerobics, even there that short retention time may not be sufficient for degradation. Typically, even with the best systems, the rates are probably an order of magnitude longer. So they're going to go untreated. Then it's a question of absorption. What part is in the water, the effluent, versus what gets stuck onto the sediment solids. Then it's a question of how are those sediment solids treated. Anaerobic digestion may, especially for the ones that are anaerobically degradable, would potentially take care of them. And then the question is, what are we doing with that residual sludge? Here, one of the biggest concerns has been antibiotic residues. And they are at much, much lower levels, but even at those levels, they drive the increase in antibiotic resistance. And so that's yet another question of low-level contamination linked to then not the enrichment of microbes that degrade them, but microbes that are resistant to those antibiotics, and then those genes jump into pathogens. So again, I don't know what's going to be the best treatment option. Right now, we're also still trying to get the basics of what impacts their faced, even if they're untreated, once they're in a watershed.
Regarding the antibacterials, are you finding or are you looking at the suppression of the bacteria in wastewater treatment that breaks down the sewage, suppression by antibacterial residues? Or?
The compounds there are probably so low, they're going to have minimal effect in terms of inhibiting growth. But still, when there's plenty of evidence showing even that those low levels are increasing the prevalence of antibiotic-resistant genes. But typically, no. I mean, the concentrations are so much below the therapeutic doses that they have minimal effect on overall growth of the community, but clearly are already selecting for distinct members within that.
Your work is on the anaerobic process, but do you know about any applications of ozone to enhance the process?
So for chemical degradation, yes, there are various, both sense and chemistry, ozonation that can break down organics. I think there the biggest issue is how much other organic material do you have, because that's going to compete with the ozonation process. And I know some of the compounds definitely can be broken down by a very strong ozonation. But again, that's no longer microbial, because that ozone is also going to kill any microbes in the system. So I stay away from it.
You showed different rates of degradation in different production environments.
Mhm.
Do you have a comparison on like base rates for what would normally be something that these bacteria are digesting? I mean, is this high? Is this low? Is this normal?
There isn't really that much data, I mean, on these sort of compounds on the face, and again,
I would assume it's a pretty slow environment.
yes,
If I was taking
compared to other compounds that we've studied, phenols, benzoic acids, so other aromatic compounds, these rates are quite comparable.
So in other words, we can consider this to be a sort of, well, we don't know, but this is like a normal rate.
This is a normal rate for a somewhat recalcitrant molecule. So, I mean, even phenols, benzoates, benzoic acid that we use as a preservative in food is not degraded as quickly as the sugar. But these rates are somewhat in the same order magnitude what we've seen for other similar aromatic molecules.
We have a question, I think we have a question or two up there.
What are some plants or otherwise chemical compounds in stormwater? Well, so to me, I mean, they would be the same. I mean, you have largely, you have your stormwater mixed with untreated sewage, and that's the overflow problem. So whatever you have in your sewage gets into the CSO. The other molecules are actually a lot of hydrocarbons, petroleum derived and so on, that are from traffic and so on that comes in, and that's then an additional issue. LC50 value of the first compound. Oh, that was comparing the LC50 of bisphenol A, monomethyl and dimethyl ester. I would have to go back on there. We can go in. There were variations on immediate and acute, so I think we can check on that. And then 94% of drugs and PCPs are excreted through urine as opposed to solid waste. Well, that depends on the use. I mean, if we're taking antibiotic or another drug, then it's a question of how are we excreting it. And those are going through sewage. And I think that's actually for most, if you think of both human use, agricultural use, all of that is going into aquatic environments. And solid waste would actually be unused drugs that would be put into the trash. And that's a minimal amount of the total drug use. I don't have those exact numbers, but again, from the large volume, it seems very much going through use. There's one study that I know of from a hospital in Switzerland where they monitored PPCP use by the patient, and they had actually their own wastewater treatment plant. And those numbers jive exactly.
We've got a clarification there.
Oh, urine versus feces. Ends up in the same place. From my perspective, yeah.
Do we need to go back to your presentation to address that other question, or?
We can. I mean, I have to admit that.
Or is it worth?
Okay.
Okay.
I mean, it's a study, so you can take a look at that in Environmental Sciences Technology by Jessica McCormick and co-authors that then goes into it. I mean, there were slight variations between them, and also looking at some of the metabolic pathways that were induced, that some went up with a methyl group, some went down, then with the two methyl groups it was slightly different. So it was an aspect that we don't actually still yet know the details of how that's going to impact the developing embryo. But clearly there were things that it was not innocuous, and in some cases had more deleterious effects as well.
Do you recommend that when the FDA does okay on a pharmaceutical product, that it check out the end-of-life cycle for faith after it's used by humans?
Yes, I think that would be reasonable to be part of actually any chemical use in terms of thinking of what's happening to it. I mean, I think we have enough notorious examples from the past that we should learn to think about our regulations to not just examine, efficacy and direct toxicity, but also safe and in different environments. And for many chemicals, that of course is part of the assays, but clearly we often don't know enough yet about them.
Are you finding caffeine to have any special effects on the biota? I mean, that was one of those chemicals that you.
There are plenty of cartoons showing overstimulated fish in their class. This is the other flip side of our concerns of emerging contaminants. Our analytical methods are improving all the time. So we're detecting more and more things. And are we concerned with them because we can detect them, or because, we know that there is a direct effect? And very often we're concerned because we can detect them and we actually don't yet know what the long-term effects are. With caffeine, I don't know, I haven't really seen anything directly, and I've been thinking of actually adding it to our degradation assays as actually having a good control of a natural compound, and so that could actually address your question of, if we relate degradation to other similar compounds. And that's actually very, very useful to do that.
One of the interesting things about your work is, where you get your data and where you take your samples.
Mm-hmm.
And with two things happening in New York City right now, both the revisiting of the Hudson River Estuary and Sanctuary Plan and the long-term control plan for the combined sewer system, do you have suggestions for how, now that Bo's not in the room, the city should think more site-specifically or longitudinally about where locations could be designated for the next 100 years to take samples in the same spot so that, data like yours can be monitored over time?
So one of the hotspots for biological activity are salt marshes. We've studied previously works with polycyclic aromatic hydrocarbons and so on, where due to complex effects of plant microbe interactions, there's often an enhanced activity and degradation of a diverse set of organic compounds in a rhizosphere zone. So, and that's of course well used also in basically wetland treatment of sewage that can be very effective as well. So the combination of the plant uptake of nutrients as well as enhanced microbial population in the root zone that can enhance degradation of all the different micropollutants. So that might be one way of thinking, okay, well, if we do have overflow, will it discharge, for example, into wetland, either constructed or semi-natural? Well, there's probably no natural wetland left here, but thinking of how to use this as a way of polishing efforts. And in many places, after regular sewage treatment, that effluent may be polished through constructed wetlands as well. And so this is one way of thinking, putting my microbiologist hat, where would I find the highest activity?
So if you have no natural wetlands almost around anywhere in Manhattan, and the only natural wetlands on the Upper Bay are about to have 3 holes of a golf course built on top of that, where else in the immediate vicinity for educational purposes, for, you know, public engagement purposes. Can you go back to Vestry Street? You have your sweet spot right there where you go down and get your bucket full of gook.
Well, right now, I could go just about anywhere. These sediments are full of the microorganisms. So that's, and then we've seen that also with other halogenated molecules that we've been studying, chlorinated, brominated benzene. The microbes are there. Then the question is, how is that specific environmental condition either conducive or inhibitory to their activity? Sulfates actually plays a big role. It's great for some compounds, really bad for others. And for example, dechlorinated bacteria are typically inhibited by sulfates. So if you have the seawater intrusion, that shuts down PCB dechlorination. But clearly it improves degradation of many of the organic molecules that have been oxidized to CO2. So that's the aspect of figuring out this combination of chemical insults and which conditions would be useful. Again, one of the aspects of a wetland is that these redox environments are continuously changing, partly with the growth of the plant, can pump oxygen into the rhizosphere zone, but that would be one reason also why the activity is much higher.
I would just point out that the huge difference between constructed wetlands and more natural wetlands, they're not truly natural, is the layer of peat. The constructed wetlands, it takes hundreds and thousands of years to develop a substantial peat layer, but even the degraded natural wetlands in our area have considerable amount of peat.
I mean, we've shown, for example, tetrabromobisphenol A dechlorination being enhanced in the rhizosphere of Spartina and Phragmites as one example, and the same with polycyclic aromatic hydrocarbon degradation. So, but again, those are often very unspecific, unrecognized what's really going on, other than often just a boost of the microbial community numbers by sometimes 2 or 3 orders in magnitude. And so then by accident, you happen to have a boost in some of the degrader populations as well.
Okay. Well, thank you, Matt. A couple of points of business. Our next lecture is going to be March 6th. We're going to hear from Dave Secor from the University of Maryland about climate changes and the effect on Hudson River white perch. And those of you that may have showed up here today without RSVPing, if we don't have your name and email, please leave it. I think we have a sign-up sheet at the front counter, because we'd like to stay in touch with you and let you know about future events. So thank you for coming, and hope to see you next month.